Okay, welcome everyone to today's session of proteins and protein folding, now in its 7th installment. So, since we also have some new people today, I would like to start off by reminding you how the whole, workshop is supposed to kind of work out, and how your setup is supposed to be. So, what I was imagining is that you optimally have two screens in front of you, right? So one would be your laptop and the other one would be your phone. Your phone is basically for watching the stream on YouTube, and you would be basically working along and, for instance, using Pymol on your laptop. This can also work if you, for instance, have two screens, like for instance your laptop hooked up to a second screen. Or, I mean, it's also possible if you only have a laptop and you can like switch back and forth, right? But sometimes it's helpful to see the information from the stream and from the presentation that I'm showing you to know what's going on. All right, so let me ask first, who's going to use Pymol today to work along? I'm going to give you a little bit time to reply. And I also want to remind you that when you have questions, feel free to basically write them in the chat anytime. I'll see if there's a comment, and then I will look at it and try to answer it. Although there is going to be definitely some delay, because somehow the whole streaming process takes like maybe 10-15 seconds to catch up. Okay, nobody's using Pymol today? Okay, that's fine. That also works. So let me then start and continue today by a little bit reviewing what we did last time. So if you remember, I mean, last time is now 2 weeks ago. Last time we kind of finished the chapter about motion, right? We talked about how basically flagella work. And although it is not 100% known now how it works, there are some ideas how basically the proton gradient is able to turn what you can see, the C-ring by channeling them through mod A and mod B, what we talked about last time. And by this, it then turns the whole hook fragment, which is anchored in the plasma membrane and the cell wall of bacteria, and then causing a rotation, thereby propelling the bacterium through the medium. We also talked a little bit about where this energy comes from that creates this rotation. I just talked about the protein gradient, which is basically like potential energy. We will see this is also important for what we're going to talk about today. So if there are lots of hydrogen ions on one side, this basically is like a separation of energy states, right? So there is a certain energy that is necessary for concentrating them on one side, right? So very similar to if you were to think about pumping water up a hill, right? So it takes a lot of energy to give it this potential energy, which then if you let it run down the hill is released again in kinetic energy. And in the same way, we looked at how basically this gradient, where basically hydrogen ions are flowing through this kind of tunnel then creates the energy to turn this whole flagellum motor construct. Right? Very, very similar like it is in a generator, which is used for hydroelectricity. And especially we looked at ATPases, right? So maybe this one. This is basically like the turbine in a hydroelectric power plant, where you have a turning part where basically the hydrogens are channeled through, which then captures the energy and turns it into ATP. And we talked a little bit about the direct mechanism of how that works, right? If you imagine this rotating part as a dissection and looking on it from the top, you have this part basically where the hydrogen or the protons go in, and then they turn around in like a carousel and then going out, right? So basically being transported through the plasma membrane, and this energy from the transport is then turned into rotational energy, which in turn is turned into ATP, which we know is an energy-rich molecule. Okay, so, and we didn't completely finish that last time, so we talked a little bit, we looked at the structure, we talked about the function of the rotation and this kind of squeezing mechanism, how it's all held in place by this kind of stator. And today I want to give you also a little bit of context. For instance, why is that a protein that we talked about? Why is that something that's like relevant for us or for life in general? And in general, this protein is one of the key components that make life possible. And I want to show you a little bit why and where in the big picture of an organism is this protein situated, and how does it make it so important? For instance, it is located in one of the main one of the main reactions that make life on Earth possible, which is photosynthesis, right? So capturing light energy and turning CO2 into oxygen, and thereby producing energy-rich molecules like carbohydrates. If we look more in detail into this reaction, I'm not sure how much you've learned about it so far, but I definitely know that you know that water is necessary, CO2 is necessary, and that sugar and oxygen is produced. This all happens in two different reaction cycles. One that turns water into oxygen is called the light reaction and uses the light energy, and it produces two molecules. One is ATP, right, the very important energy currency, and the other one is NADPH. And how that energy is used, that's produced in the light reaction, is it is turned into sugar by fixating CO2 in what is called the Calvin cycle. Right, so definitely during high school you will have more detailed knowledge and exercises about this whole process which is going on in plants. So, and I hope maybe you have already talked about it, but it's definitely something that's very important for us, right? Because turning light energy into sugar makes sure that things can actually grow on planet Earth. So if we go even more into detail, especially in this energy producing step, right, in this light reaction, what we can see then, there are different proteins involved. Right? So These proteins can be mainly found in chloroplasts. As you know, chloroplasts are these like pill-shaped organelles which are found in all plant cells and which make them look green in the end. And in these chloroplasts, you have what is called the photosystem. Photosystem 1 and 2, and at the end is the protein that we talked about last time. So, and how this whole thing works, and how it turns basically light into ATP, is shown in this kind of complicated looking reaction. But I don't want to go into detail about everything. I just want to tell you the main hallmarks of this, right? So this all involves the transfer of electrons within these proteins, which then turn ADP plus into ADPH, which is also an energy-rich molecule. And while this is happening, you can see here are also protons, right? The protons, while the whole chain is transporting this electron, they transport protons from the stroma of the chloroplast into the thylakoid lumen, what it's called. Like, thylakoid is one of those stack-shaped like membrane stacks inside chloroplasts. So inside the thylakoid, you will have a high concentration of protons. And as we talked about last time, the protein that we looked at uses this difference, or this energy potential that is caused by the difference of concentration of protons, to turn this into ATP, right? So by going back through the membrane and turning the ATP synthase, we produce ATP. So one of the very, very fundamental processes in life, in organisms on this planet, relies heavily on the ATP synthase. So that is one part that I wanted to show you. And also for the other part, it's super, super essential, right? Maybe you've seen the picture on the left before. I hope you have. It's called a food pyramid, right? So if we think about ecosystem, plants turn light energy into energy-rich molecules, and are then the basis for other organisms that are on top of that consume them, right? So plants are what's called the producers, and the rest are the consumers. They basically make use of this captured energy by the plants. So we talked about how the ATP synthase is very important in producers, but it's also super, super essential for consumers, right? Primary, secondary, tertiary, right? Because also we ourselves, we are consumers. Not in an economic type of thinking, but in a biological way of thinking. So we eat natural products, right? So like fruits, vegetables, and also other animals. And what we get from those is, okay, here's proteins, carbohydrates, and fats. So those are all energy-rich molecules which we can then use to turn ADP into ATP. Right? So to produce basically something that is useful for cells to, for instance, exist, or just multiply, make other cells, make other cool proteins, and so on, to just exist. And this process is called metabolism, right? And in detail, it gets more complicated, and I don't want to get into the whole process of it. But I mean, the simplest form of energy-rich molecules is probably glucose, right? And glucose is super, super important for every cell in your body. And the whole purpose of metabolism, or of this whole process, is to turn glucose into, down here, ATP. So there are different things like glycolysis, pyruvate oxidation, and the citric acid cycle. And finally, we have the oxidative phosphorylation. So it a little bit depends on the organism which we're looking at. This is, for instance, exemplary for humans, right? Or animals and other things. They have all three. Well, sometimes some organisms, they just stop at like number 1 and number 2. And for them, the number 4 and number 3 is not so important. But yeah, we don't want to go in too much into detail with that. Important is number 4, right? So, for instance, in humans. Again, you see there are several, or like a row, there's a row of different proteins, which, in this case, it's the other way around. So before we had NADP plus turns into NADPH. In this case, it's the energy-rich form, NADH, turns into NAD+, and thereby transferring an electron, which turns oxygen into water. And during transfer of this electron, what happens is, again, protons get transferred. They get transferred into an enclosed space, where the concentration gets higher, and you see where this is going. This concentration difference is then used again by the ATP synthase to turn ADP into ATP, an energy-rich molecule. So, in whatever way you look at it, the ADP synthase is super, super important for consumers, producers, for life in general, as we know it, right? And to think about it, that this essential protein has such a, well, I don't want to say simple, but very, very intuitive mechanism that we talked about last time, that is so similar to what we know as a water turbine, is, I think, very, very fascinating. And that's also the reason why I wanted to show it to you. Okay, so kind of, with that, I wanted to show you basically the importance of what we looked at last time. And now I think we can move on to our next topic in this workshop, which is going to be transport proteins. Right? So, transport proteins, let's see. I want you to imagine what a cell membrane is a little bit, right? So probably from biology, you've had to memorize at some point the different parts of a cell, right? And maybe also their function. And what usually comes up when it comes to the cell membrane, well, the cell membrane is a layer that separates the inside from the outside. Right? And this layer is actually like not permissible. So usually things don't go through it easily. Of course, there are some things that can go through, but for the most part, molecules don't really go in and out of cells. Right? Generally, like free flow of molecules from inside and outside of the cell would not be a good thing. So, but the truth is, things have to come in and things have to go out of the cell, right? For instance, if a cell wants to live, energy-rich molecules have to go in, right? Like on the left side. If a cell wants to, or needs to use energy, it needs, for instance, sugar, right? And this sugar somehow needs to get inside the cell. And if the cell produces something, right? For instance, like here, like hormones, these products then need to go out of the cell. So this is actually a very, very important function that is taken on by transport proteins, right? So, because if you think about it, if you were just to poke holes into this in the plasma membrane, just everything could come in and everything could go out, which is not something that's very desirable. Because maybe also things that you want to keep in would be able to just fly out and go away. So the cell needs like a selective process to kind of say, okay, these are things that I want in, and these are the things that I want out. So, and transport proteins have a very essential function, or this essential function. Okay, so if we think about this kind of transport, we've kind of looked at proteins that would be able to have this function in past workshops, right? So in one of the very, very first ones, when we talked about secondary structures, I already talked about domains, that part of proteins that have a specific function, right? And we talked about, for instance, this one, which was a beta barrel, which is often found in so-called transmembrane proteins, right? So parts of proteins that can be embedded into the plasma membrane to then form some kind of pore, right? So because the beta barrel, as you remember, well, it looks like a barrel. It's barrel shaped, and it would basically make a hole into a plasma membrane if it is embedded. We also have other similar domains that can be embedded in the plasma membrane, which usually are built out of different alpha helices. Also usually arranged in some kind of barrel or circle resembling shape. Okay, so, and most of these transport proteins, which we're going to look at, have very similar features like this. Okay, so, and if we think about transport proteins, I'm just going to tell you there are basically two different kinds. Very, like, not okay, two different groups, I would say. So ones that work actively and those that work passively. Okay, what that means, we're going to have a look right now. So first I want to go into the detail a little bit how passive ones work. So passive ones basically work according to the concentration gradient, which means maybe you know from physics or chemistry. If not, actually let me skip to the next slide. Similar to what we talked about the proton gradient is basically true about all molecules, right? It's a little bit hard to imagine, perhaps. But if we look at this setup, right? So here we have a U-shaped glass tube, and it has water inside of it. In the beginning, maybe if you just don't look at the dots on the right side, let's just say we have water in there. So both levels of the water surfaces would be even, right? Would both be at the same height. Now, what we do is we put a semi-permeable membrane in between it. Semi-permeable meaning that only certain things can go through, right? In this case, it is water. So the only thing that can pass through are water molecules from one side to the other. Now, what has been done in this case is on side 2, they have basically put in an impermeant substance, so something that cannot go through this membrane. Could be any molecule. Could be salt, could be sugar, could be anything that's like miscible in water. So what that does is it produces a concentration gradient. And what you can see, and maybe if you want to, if it's hard to think about it, we can definitely do it also in Mannei as an experiment. What will happen is water will want to dilute the gradient on the right. So it will transfer from the left, from side 1 to side B, and thereby changing the height of both surfaces. So one's going down and the other one is going up. So basically, in this case, that is called osmotic and hydrostatic pressure. So the osmotic pressure is transferred into a kind of potential energy of, well, that is related to gravity. Similar to what we looked at before, what I talked to you about is the transfer of water up and down a hill. Right? If you transfer water up, you have potential energy that you can then reverse into kinetic energy again if it goes down. So the same thing, the same principle is happening in this U-shaped tube. Right? The concentration is high on this side, so the water wants to dilute the concentration and make it equal to the left side. That's why there's a certain pressure of the water to go to the right side. And this is where the whole energy from this passive transport comes from. So if you go back, on the left side, you basically, the semi-impermeable membrane is the plasma membrane. You have sugar molecules on the outside, for instance, and you don't have any on the inside. So that means you have a gradient. You have osmotic pressure. So on the one side, water wants to go out and dilute the sugar, and on the other hand, sugar wants to go in and even out the concentration. And this can be easily facilitated by using these kind of pores, right? These kind of proteins that form holes and then transport this molecule from one side to the other. Right? So it can be as simple as a hole. There are proteins that are called aquaporins that are basically there to facilitate the water flow through the plasma membrane, which is not so usually, what is not so easy usually. Yes. And those are also usually not only holes, but also selective ones. So there are transport proteins that are only responsible for transporting, for instance, glucose, sugar, and nothing else. So they have specific shapes that only let through the sugar molecules. A different type of these passive transporters are called symporters. So they don't only transport one molecule, but they kind of have interdependent transport of two molecules. So, for instance, one sugar is transported with like a fat molecule, something like that, right? And they only can go through if they go through at the same time. Or there are also concepts like antiporters. In this case, something goes in, but at the same time, something else has to go out. So these are the basic concepts, how passive transport works, and what kind of variations there are among different transport proteins. Today, we're not going to look at those. I'm not sure that there are many, many different kinds, and many are, I would say, important in their own respect for what they do and what kind of molecule they transport through. But generally, there are more interesting molecules to look at. And today I would mainly want to focus on the active transport, because it also kind of ties into what we looked at last time. So in this case, well, active means using energy. Energy in the cell is usually, like 90% of the time, it's ATP being used and split into ADP and P. And active transport is special because it can transport molecules against the gradient, right? So in this case, if you look at the right picture, there are many molecules on the right and few on the left. So if we were passive transport, it would actually go from right to left if it were just a hole. But since it is active transport, even those two molecules on the left, under the use of energy, will be transported to the right to increase the concentration. And this is a very, very important process in cells because if you think about it, for instance, also many molecules that are used for energy are very, very essential. If you were just to, let's see, if you think about it, if you were to eat, let's say 10 grams of sugar, right? And if you think about this membrane, and inside outside would be, for instance, inside your stomach and inside the cell, if it only were to equalize the concentration, you would basically take up 5 grams, and 5 grams of sugar would stay inside your stomach and would not move into your cells, right? So if you would not move these sugar molecules against the gradient into the cells, you would lose a lot of energy that you could otherwise use for your cells and for your body, right? So this active process is super, super necessary to be energy efficient also for your body and how you use food, for example. But today we want to focus on a little bit a different example where transport is super, super important. And it involves both active and passive transport. So today we're first going to talk about the active one, and then probably next workshop we're going to talk about the passive transport, which is happening. And I hope you kind of can already tell what kind of cell that is. It is a nerve cell, right? So basically today I want to tell you about one part, how basically nerve cells work. So we will, well, not quite now, but maybe I hope that after this and maybe next workshop, you will kind of understand how nerve cells can transmit signals and how this relies on transport proteins. And I know this can be kind of intimidating and it can be a little bit difficult at first, but it's, I think, a super, super interesting process that's going on every day and is super important for all kinds of living organisms. And understanding this kind of process can give you a lot of insight about how your brain works and what its limitations are. So without further ado, let me give you a short kind of conceptual look at how nerves work, right? So we all know that nerves, they transport signals, right? Signals, in this case, we think about it as kind of electricity. So what's electricity? Electricity is basically a difference in potential. it's even similar to the gradient that we just talked about. On one side, there's more of something than on the other side. In this case, one side and the other side means plus and minus. On the minus side, there are more negative molecules than on the positive side. Right? So there is a tendency in nature, which is very basic physical concept, that wants to create an equilibrium between positive and negative and make it zero. And we've learned how to use this difference to build actually electrical machines and appliances and devices. Right? Everything we use that is connected to electricity uses this concept of difference of potential. Right? So in order for electricity to flow, you need a difference of positive and negative potential, right? And this is a little bit, or well, not a little bit, very similar to how nerves work. So, but nerves are also a little bit different. So if you think about electricity is electrons moving because of this potential difference. Right? In nerve cells, what is actually moving is not electrons. It is the actual potential difference, or the difference of the potential. Right? So what you see on the picture on the right is, on the bottom is basically like a one part of a nerve, right? Which is called the axon, which connects one nerve to another. So what you have in blue on the bottom and on the top is a plasma membrane. And so now what you have indicated on this plasma membrane is actually potential, the plus and the minus, as if you were to look at a battery. And this plus and minus travels along the plasma membrane to another cell. Right? So usually you have a positive potential on the outside and a negative potential on the inside. And once a signal comes, this potential is inverted for a very short time. And then inverts on the next and the next and the next and moves forward like that. So this whole process we will look at more in detail next time. Right now, what I want you to understand first is like a nerve works a little bit like a loaded gun. Right? So initially we already have a difference in potential. Right? So the plasma membrane is already charged into plus and minus, into difference. So once a signal comes, it's like if you were to pull the trigger of a gun, then it shoots off and reverses the signal, the potential, and then thereby transmits the signal. So today we're going to look at how this gun is loaded, if you want to look at it like that. Right? So how this initial positive and negative difference is created. And something that you can already see in this picture a little bit is that this involves the transport of proteins through the membrane. One being potassium, and the other one sodium. So what we're going to look at today is called the sodium-potassium ATPase. And if you've been here the last time, I hope you kind of already know what the word means, and it kind of shows you and tells you how it works. Right? Because ATP is what we talked about, is the currency of the cell, and the A's at the end, the ending, it means basically that it splits ATP to use its energy. So because sodium, potassium, the two ions, and I told you it's a transporter, we're going to look at the protein that transports sodium and potassium through the membrane by using ATP. Also part of the family of ATPases, which we looked at last time, right? So last time, the molecule that we actually looked at was not the flagellum, but it was the ATPase that we looked at and kind of tried to understand a little bit more about its mechanism. So, coming to, I know today's theoretical part was pretty long, but I hope it was still interesting. So now let's go a little bit more into the hands-on part of today's workshop. Since I have not gotten any feedback yet if somebody is using Pymol, please let me know if you do while I get set up, and I will go slower. If nobody's using Pymol, I will just pretend that nobody is, and I will go according to the speed that I would be working at. Okay, so let me switch to Pymol. Okay. Actually, okay, if you're using Pymol, it's for the first time, it's a very, very useful tool to look at proteins in 3D. It works. Okay. To look at proteins in 3D. Right? So, as you see, maybe if you are new to this workshop, I would recommend you go back actually to the first part of this workshop, where we talk about a little bit about the structure of proteins and also a little bit how to use Pymol and what Pymol is actually for. In general, it's just to look at the structure of proteins and thereby understanding their function. So basically, this is what we want to do in this workshop. So today, we will look at two models of proteins. One of them was 4-HYT. So we're just going to type in fetch 4-HYT. And what that will do is it will get a model from a protein database server. And it will display it very with all its information and in the basic representation, which is the cartoon with like the ribbon type of structures and the beta sheets and whatnot. We are also going to load a second molecule, which is basically the same, but in a different conformation. Yeah, but let's do that in a second. Okay. One more time. The code is 4-HYT. Okay, and now you can see basically this protein, and actually you can see two parts of this protein. First of all, I would like to work a little bit with it, just removing the extra pair. So we're going to display the sequence as always in the top toolbar. Go to sequence and just click on it. And it will display this long row of, which represents the amino acid sequence of the protein. And since there are two, we probably have to start somewhere in the middle to delete the second half. I will just say from now, I think that's it. EA. Yeah. I actually found out how to select amino acids a little bit easier. All you have to do is basically shift and left click to select all until this point. And then it will look like this. Selection always means that these pink type of dots appear. So we're going to delete the first one, the first half of the amino acid sequence, by just selecting it. And in the amino acid sequence, we're clicking right click and then remove. Right? So what you're left with is one part of the protein and some extra leftovers, which we also would need to clean up. Extra leftovers you can see in the back of the protein, of the sequence. We don't need to worry about those at the moment, but you can also just click them to select them. That's all. Should be more or less. Okay, and then we can also right click, remove, and then that's it. What are those? Oxygens, okay. Also remove. Right, and now we have like one protein part. So this is the sodium-potassium pump, as it's also called. Sodium-potassium pump or sodium-potassium ATPase. So similar to what we did the last couple of times, what I would like you to do is first take a look at it. Right? Take a look at it and try to guess what subunits there are. For today, for this one, I actually have to admit it's going to be a little bit more difficult than the last times. This is not so completely clear if you're just looking at the structure. And it would take a little bit more time to really get an overview. But definitely, like last time, it always helps to give it a more colorful appearance. Right, so click on C for color, then spectrum and rainbow. And that gives you a lot better picture of what is happening. Okay. I don't know. If you don't like the molecules in between, you can also get rid of them. I'm going to select some of them because they're not really essential for the function. They're probably like a remainder of what was used to crystallize the protein crystal. But something you should not remove, definitely, is what's inside here, which is a sodium. Oh, wait, that's not a sodium. Right. Should be sodium. Okay, so, and like last time, I would ask you to go to the whiteboard. Oops, that's not the whiteboard. whiteboard. Strange. Just give me a second to find and show you the right window. Oh, okay. Now I got it. Don't know why I didn't get it in the beginning. All right. So, if you are on the whiteboard, again, choose the pencil, write your name, and you can also choose a color if you like. And I'm going to wait until I see some names of the people who joined the whiteboard. Okay, Pavel, you seem to like purple. That's good. Okay, somebody else. Maybe Koto. Since I know you're here now. Actually, something that I hope that we can get to work in the future is streaming through Discord. Because I mean, YouTube is great, but if we were to do it on Discord, we wouldn't have so much delay. Because right now, I mean, even if it's only 10 to 20 seconds, it still makes it very awkward to wait for everybody to hear what I'm saying and then react to my exercises. So, but not sure. Maybe people are very passive today. That's also okay. So going back to PyMol and the whiteboard. Now that you're looking at the protein, I want you to think about what different parts would you basically, or would you be able to identify looking at this? Right? So like maybe subunits that have a different functions from each other. If not, I will of course tell you anyways. For instance, I can give you a tip in the beginning. Maybe it helps if I show you the molecule, if you're not using PyMol, of course. Let's just start from the top, right? So there's definitely, I'm going to draw this on the whiteboard. There's definitely like something like a blob on the top, right? Something like a circular shaped kind of, maybe it even looks like a lid, right? Okay. Then we have something that I would draw similar to this. I mean, now I have to pretend I haven't looked at this before, but probably if I were to see this for the first time, Going back to the whiteboard, just to give you an idea of what I'm thinking about, I would think it looks something like this, right? Comparing it to the picture, like a conceptual drawing of this protein, I would draw it something like this, right? So there was like this lid type, then you had all these alpha helices, right? In bundles. I'm just gonna draw on like squiggly lines, and so on. And then some unstructured areas on the bottom, with also some better sheet type structures, right? And also unstructured on the top. Okay. Maybe zoom out. Nobody's on the whiteboard today. Unfortunate. Okay. So now, how does this thing, what I just drew, turn into a transporter? Oh, okay, very good. Thank you, Pavel. Right. So, first you have to know, like, I mean, if you're talking to the transporter, where is the plasma membrane? And Pavel correctly identified, okay, there is actually a domain, which we've seen before, that consists of kind of circular arranged alpha helices, which is a very, very common motif for transmembrane domains in proteins. So he drew the plasma membrane in with the purple lines. Very nice. Okay, good. Also, perhaps if I didn't know about this protein already, I would maybe guess that the lid has a function in regulating transport. But we'll see about this in a second. Since we know there's an ATPase in there, I would guess, looking at the structure, it's either here or here is a potential ATPase. ATPase center. Okay. All right. So when the bottom part, uses ATP and turns it into ATP and P, this somehow then changes this middle part to let through sodium or potassium. All right. And simply put, this is how it would work. So, of course, people have looked at this a lot more in detail. And how this really functions, I want to show you now. For this, I would like to go back to PyMol. Okay, very good. And I want to highlight basically 4 amino acids. Maybe, okay, it's actually hard for me to see if I don't have the presentation open. Let me write them in the chat really quick. D84, E327, E779, D369. Okay, just for me to see them. So we're going to look at these in the protein, because these are very, very essential. 804. 804D. Very good. Actually, to make work a little bit easier, because I wanted to also use a different protein model, let's first add this to PyMol. 3KDP fetch 3 okay. KDP. Right, which adds another molecule, basically the same protein, and again we have to delete half of it. Let's see from here. Yep, sounds good. Right click, move. Some things are left. This. And we can delete those. Okay. Very good. So this is going to make it a little bit easier. Because we can identify both. We can identify the amino acids in both molecules. So 804D. 804D. Here's one. 804D. Here's the other one. Okay, very good. The next one is E327. 327. 27. E here. And here. Good. Next one is E779. 79. This one. 779 and also here. And last one is D369. 60. Here's it here, but I didn't see it in the other one. So this selection we are going to rename. Click on A, rename selection, and then just going to call this amino acids. I guess, and then we're going to give them a different color. Maybe red. Okay. Actually, first I would like to focus on these amino acids and what they do. Well, we also need to kind of show them as licorice so we can actually see what they do. Okay. And those amino acids have a very essential role in this protein. And you can see why if I zoom in a little bit closer, because now here, those purple spheres, they represent sodium, right? So you can see in this model, sodium is bound to the transporter in the membrane, right? Remember, this part with the alpha helices is the transmembrane area. So where it's red, you can see that these amino acids, which are negatively charged, are there to bind the sodium and hold it in place. Basically, they're there for the affinity, to regulate the affinity of sodium to this whole protein. And you can also see a little bit that there are two ways for the sodium get in there, right? One is from the bottom, like you can see basically where the center of this kind of alpha helix barrel is, right? So this is where the molecules could come in. And there's also on the top an opening where they could come out. So how this molecule works depends a lot on modulation of this affinity to sodium and also deformation of these alpha helices to open and close these openings for the sodium to go in and out. Right? So these are 3 of the amino acids. The other one that is down here is responsible for the catalytic activity of basically getting the energy from ATP. And it's kind of getting the energy by being phosphorylated. So the phosphor from the ATP is transferred onto this glutamate, which is on the inside part of the membrane, right? And as we talked about, this phosphorylation and this catalytic activity leads to a large change in basically the structure of this whole protein, which then can define the function of the protein. Okay. And to understand this a little bit, what we're going to do is we're going to align it. Right? So like we did before, we're going to take one protein and just put it on the other one. There's a very simple command. All you have to do is you press on A, then you click on, actually, where is it? Here. You click on the line, and you have like another menu that opens, and you choose to molecule, and then 4-HYT. Okay, oh, it actually went pretty fast. And what you will get is both proteins basically stacked on top of each other. Right? And what you see here, if you follow this alignment, because I told you about those, this is the protein 2 different conformations, right? One is basically after using ATP, and the other one is before. And what is happening in this protein, you can see, well, in a large part of this transmembrane alpha helices, there's not much going on actually. They are firmly in position, I would say. Also, what I described before as a lid is not changing dramatically. Okay? So that does not seem to be very related to the function. The change that we do see is, for instance, taking place mainly here, right? In this light and dark blue part. Good question. Actually, to answer that question, Christina, is actually what helps is to look at the protein, and I'm just like the one that's open to the outside basically is after using ATP. Basically has an open tunnel that can basically let the sodium get out to the outside. And the one that is more open there is the one that used the ATP. In this case, that is okay. That would be the 4-HYT. Is the after using sodium, and the other one is before. Well, in this case, there is no ATP molecule bound, unfortunately. So, well, in this model, it is not included. I think they have aluminum fluoride. I have no idea why. I could look it up. It's basically blocking the binding site for ATP, and this is maybe an inhibitor. Who knows? Okay, very good. So we see a lot happening with the light blue and the dark blue alpha helice here. So we can expect this to move a lot between those two different states. And also we see there's a lot happening also in this light blue, dark blue part on the bottom, which is very close to the site where ATP binds. Right? So it's also, again, very similar to what we saw before in basically in some of the other molecules that we looked at, where the hydrolysis of ATP like basically initiates a conformational change, which is then kind of exaggerated over several alpha helices and changes a large part of the protein. Not all of it at that, but still. How much is changed, of course, depends on the protein and on the function. But in this case, mainly focus on these two alpha helices. And of course, there are people who have taken a look at that in detail, and I want to show you a little bit what they have found. See, let's go back to the presentation for a second. Okay. So, this is the protein that we just looked at, just a different representation. So not the cartoon view that we were looking at just now, but actually it's represented as kind of strings, and also you can see a surface representation. Highlighted with those squares are, for one, the sodium binding pockets, and also the ATP binding pocket at the bottom, right? The membrane would be where the gray, light green, or brown part is. And also what they did in the paper that I'm referring to here specifically is they did a schematic drawing like I'm always asking you to do to show how it functions, right? So you can find the different parts on the left represented in the cartoon-like representation on the right. Right, so there's a red part, a light blue part, a yellow part, and the two alpha helices that I was talking about before in the model. And then the part that doesn't change so much is just grayed out. So already here, if you look at a more detailed drawing of this like idea of how it could function, you can already see they kind of highlighted some things. For instance, like an entry tunnel, in a way, for the sodium to get in, and or like a binding pocket and a subunit that might move on to the other part. So what they were looking at is, like, for instance, if you look at the sodium binding pocket, they looked at the different states, and what they found, maybe it's a little bit hard to imagine. Here's a little bit, the surface is more pronounced, and it's looking a little bit from the top. So this is basically the tube where the sodium would come out, right? So A is before hydrolysis of ATP, right? You can see it's kind of closed up. So the highlighted amino acids and the tunnel to the outside is kind of closed up. But if you looked at E after hydrolysis, you can see it's basically open and the sodium is able to escape. So going back to the schematic representation of that, you can see, well, first, ATP binding takes place, right? So ATP goes into position. And in this conformation, the affinity to sodium is very high, to sodium ions, right? So in total, 3 sodium ions can find place in here, right? They go into this tunnel and then are fixated by certain amino acids, the one that we highlighted. What then takes place is the hydrolysis, and you see this red part kind of makes a connection with the yellow one, and thereby the yellow one kind of changes its conformation and closes up this tunnel. Then the ADP goes out and the phosphorylated glutamate stays there, of course. And that leads to another conformational change, which opens up the tunnel, which I just showed on the other slide, to let the sodium out into the other part, into the outside of the cell. Right? And if it's empty, actually in this conformation, it's not very attracted to sodium anymore. Right? It has a higher affinity for potassium. So if you have this empty state, actually potassium likes to come in and take sodium's place. Right? Just because of the structural constraints of this conformation. Well, and if sodium is bound, this leads to a basically restructuring of the whole protein and a closing of this outer tunnel. And we go back to the initial state where this inside tunnel opens, the potassium goes out, it's dephosphorylated, and another ATP can bind and another sodium can go in. Right? So this is basically facilitating, this whole protein is facilitating transport of sodium outside and potassium to the inside by sequentially opening and closing basically axes, and also changing its affinity to these specific ions. And all of this is facilitated by the hydrolysis of ATP and the use of its energy. Also to maybe make it a little bit more well, like more like a transition, I also played a little with Pymol. And something that is not included into the free version of Pymol is, of course, the morphing function. And yes, okay, it actually took a while to compute it. That's why I had to do it beforehand. And I'm just going to show you what is happening to it, right? So this is the whole protein, like before, with the highlighted amino acids, right? And you see some sodiums. And well, the morph function just basically calculates frames, like a video, between two different states, right? Basically the computer is thinking about how could I get from this conformation of the amino acids to the final one, which I define. Right? So it's not a 100% accurate thing. Sometimes there are some freaky things going on, or the alignment is not 100% correct, and so on. But if I now press play, you can see it's starting to move, right? And you can see specifically those changes that we were talking about, right? So there's not much going on up here. Of course, there's some unstructured parts that are always moving. Those will always be flexible. But a big part of the transmembrane of a helices is not changing at all. Or, well, very little, I would say. Then we have the bottom part, right? Which I think if we turn it into the conformation that it was in the schematic drawing, it would look something like this. right? So you have the bottom part with the ATP, and you see that basically this part and this part make contact. And through this contact, there's a big change happening in these two alpha helices. Right? Like here it even looks like an unstructured part becomes into, or changes shape into part of a alpha helix. Alpha helix. So and if you look at it from the top, right here it looks like it's being pulled down a little bit. If you look from the top, if you concentrate on the sodium, you will see that actually it's opening up. Okay. So this tunnel to the outside is basically open up for the sodium to go out. While on the other hand, that's actually not visible as much. Yeah, maybe that's not the conformational change between those two states, but definitely the opening of the outside part. Okay. Very good. Let's see. Then let's go back to the presentation. So this all seems very, very complicated in a way, although, well, if you think about what's actually going on, it's not that complicated per se. It's a simple structural change, which changes basically like opening and closing of tunnels. Right? And simply put, instead of like maybe remembering this whole complicated process, let's just, as a final summary, kind of just watch this video, and it kind of repeats in a way what this whole process is, how it works.
Pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, very good. So just to simplify and to summarize everything, this pump is very, very essential for many things happening especially also in the human body, right? So finally, I just want to like give you, now that we've gone into detail so much, also go back a little bit to give you the context of this whole process that I just described to you. Right? Because we talked about, I want to show you in the end how neurons work. Right? I talked about that neurons work because there is a electric potential between the outside and the inside of the membrane in a neuron and an axon. Right? So the outside is positive and the inside is negative. In turn, this is facilitated, this is achieved by this sodium-potassium pump. right? So in order to build up this gradient and this potential that can then be released to transmit a signal, what is necessary is to pump out 3 sodium and pump in 2 potassium, right? So, and if you think about it mathematically, you put 2 plus in and 3 plus out. So logically, in the end, you will end up with more plus on the outside than on the inside, right? But there are some other factors that also make up this electrical potential. Okay. And with that, I would like to end today's workshop. Of course, if you have questions, I'm open to answer them, and I hope you have some, because today was maybe not the easiest of the workshops so far, because a lot of physics and biology come together here, right? And if you haven't had biology or physics, or are not very interested in either of them, maybe following might be a little bit difficult. But I try to at least make it understandable for everyone. Okay, so I'll give you a little bit of time. And if this is the time when you decide to go, thank you for coming. And we'll see you hopefully next week, when we'll then talk about voltage gated channels, right? So basically these transporters that are essential for actually making this depolarization and making the signal progress along the axon. Okay, yeah, so I'm open for questions now. Okay, good question, Pavel. Although I thought I mentioned that. So yes, so logically, moving 3 plus outside and 2 inside means that there will be more plus on the outside, thereby creating this electrical potential of having more plus outside than inside. So outside becomes plus and inside becomes minus. Okay. So I hope I'm not cutting anyone off and everybody has caught up with the video and the delay. But if there are no more questions, again, thanks for coming. And always, maybe you don't have a question now. And maybe if you think about it again, or maybe you have not only questions, but also things that you would like to know more about, you can also, of course, talk to me always on Discord or Slack or wherever. I'm glad to answer any questions regarding that, because I think it's very fascinating how all this stuff works. All right, then see you guys next time. Bye bye.